Welcome, everybody, to this episode of Facebook Live, where we explore the intersection between Christianity and science. My name is Dr. Fuz Rana. I'm a biochemist, I'm a Christian apologist, and I'm also the Vice President of Research and Apologetics at an organization called Reasons to Believe. If you want to know more about our organization and the types of resources that we have available for you, please go to our website, www.reasons.org, and check us out. Hey, today I'm going to take on a really controversial question that relates to the origin of the SARS-2 coronavirus. Was the SARS-2 coronavirus genetically engineered in a laboratory? If you spend any time online at all reading about the COVID-19 pandemic, I'm sure you've come across the claims on the part of some people that this virus that causes the COVID-19 pandemic, SARS-2 coronavirus, actually was created in a laboratory as opposed to originating through microevolutionary processes out of a natural reservoir of viruses that would infect animals like bats or the penguins. And so we're going to take on this question today, but instead of taking an approach that you might anticipate I would take, I'd actually like to use this whole controversy about the origin of the SARS-2 coronavirus to address another controversial question, and that is related to the nature of intelligent design. Is intelligent design part of the scientific construct? So we're going to take on these two questions today, but before we do that, just a couple of minutes for some business, if you will, some show business. I would really love it if you would take a moment to check in the comment section. Say hi, let me know who you are, where you're watching from. Whether you're watching this as a recording or as a live stream, this is really valuable information for us to have as we plan future broadcasts, but it's also a great way for me to get to know who you are. I really want to have a sense of who is watching these programs, who is interested in these science-based conversations, and why you're interested in learning more about how science and Christianity interrelate. Also, use the comment section as an opportunity to offer your perspective on these two questions. Was the SARS-2 coronavirus genetically engineered, or did it arise as a result of microevolutionary processes and zoonotic transfer into the human population? Or do you think intelligent design is science? Why or why not? And then last but not least, please take a moment and suggest topics or questions for future broadcasts. I want you to know that I don't have time to respond to every comment, but I read every comment and I very seriously consider what you have to say. And indeed, future broadcasts are shaped and dictated by your comments, your suggestions, or it influences the types of web articles I write, or even content that I might present in talks and what have you. So your feedback is really much appreciated and is really very valuable. Okay, without any further ado, let's take on this question. Was the SARS-2 coronavirus genetically engineered in a laboratory? Now, the part of the argument for why people think that the SARS-2 coronavirus may be a human construct as opposed to the product of zoonotic transfer through evolutionary mechanisms has to do with the fact that the virus first appears, patient 0 was living in Wuhan, China. And Wuhan also houses a biotechnology lab that has been involved in work on coronaviruses, as well as has been involved in what it's called gain-of-function research. Now, if you're not familiar with what gain-of-function research is, it's actually part of a larger program conducted by virologists as well as other microbiologists who try to deliberately, in a laboratory setting, increase the pathogenicity and the infectivity of microorganisms like viruses, with the idea that if we can understand what kinds of genetic changes take place that lead to the virus is becoming more virulent, much more infectious, much more likely to be transmitted from host to host, that we can gain fundamental understanding into the biology of, again, virus infectivity, transmission, and pathogenesis, which can lead to biomedical treatments or ways to maybe help to control or curtail future outbreaks of viruses like the COVID-19 pandemic that we're currently struggling with. Now, the reason why researchers, again, want to do this gain of function work is primarily for the basic science understanding, but there's some people that are doing this gain of function work as a way to develop biotechnologies. For example, could you genetically engineer a virus that would infect, let's say, rodents and make it highly transmittable, highly infectious, and much more deadly? This would be a great way to control pest populations, as an example of a biotechnology application that could emerge out of gain of function research. Another reason why people are interested in this work is for biosecurity purposes. There's the concern that bioterrorists may genetically engineer microorganisms, including viruses, as, again, bioweapons. And so by understanding the types of technology required and the types of genetic changes required for, again, increasing the virulence of, let's say, a particular strain of a virus, that, again, gives insight that could have biosecurity ramifications. Now, I think it goes without saying that this kind of work is really very risky work. And in fact, there are a number of government organizations that have actually spoken out against this type of work. So, for example, as I understand it, in 2014, the White House Office of Science and Technology actually halted federal funding to gain of function research programs. And so for people that are suggesting that maybe the SARS-2 coronavirus actually was a genetically engineered construct that was created in a laboratory, this is not necessarily an unreasonable question to ask, right? Because again, you have patient 0 appearing in Wuhan. Wuhan, again, hosts this biotechnology laboratory that was working on coronaviruses and also working on gain of function research. So I don't think it's unreasonable to think, could this actually have been a laboratory construct? Now, the case at this juncture is largely circumstantial, and you could argue it's just simply an unhappy coincidence that these correlations exist. But in order to really settle the question, you want to do a detailed analysis of the SARS-2 coronavirus genome, looking for signatures in the genome that would suggest features of the genome that look as if they were non-natural in their origin, that look as if they were intelligently designed. And so researchers have actually done this work. In fact, in 2019, in Nature Medicine, towards the end of 2019, there was a correspondence piece published by a large team of collaborators that actually looked at the origin of the SARS-2 coronavirus, and as part of that work, actually addressed the question as to whether or not the SARS-2 coronavirus had an origin in a laboratory setting. So the point that I want to make here, first and foremost, is that that by virtue of publishing that paper, this is an implicit acknowledgement that intelligent design is literally part of the scientific construct, because these researchers were essentially making a discrimination between whether or not the genetic composition of this virus, the structure of its genome, looked as if it was arising through natural process mechanisms or through the deliberate activity of molecular biologists who might have genetically engineered this virus. So again, that is an implicit admission that, again, intelligent design is part of the scientific construct. But I thought it would be a little bit fun to just spend some time looking at specifically the details of the argument presented by people that think the SARS-2 coronavirus is actually a genetically engineered construct, and those in the scientific community who have argued that it is actually the outworkings of evolutionary processes and zoonotic transfer. Now, to be frank, I had very little interest in this question. I've seen, again, these claims that the SARS-2 coronavirus was a genetically engineered construct. To be blunt, I dismiss them as being ludicrous claims, part of some kind of, bizarre conspiracy theory, that there really was not much to this argument whatsoever. But a few days ago, I was involved on the sidelines watching an email conversation among people that I actually have a high level of respect for, as scholars, as people that think critically, and a number of them were actually entertaining the possibility that the SARS-2 coronavirus was a genetically engineered creation, that it was made in a laboratory. And the whole conversation centered around a YouTube video that was published on May 4th, 2020 by a guy by the name of Dr. Chris Martenson, who apparently for the last 120 days or so, every day has been going to YouTube, posting videos about the COVID-19 pandemic and the SARS-2 coronavirus. And according to the stories that I hear, Dr. Chris Martenson was among those scientists who argued that the SARS-2 coronavirus originated as a result of zoonotic transfer. But recently, he's changed his mind. And in this particular video, again, that's entitled, let me see here if I can find the precise title, Lying Virologist and Self-Inflicted Wounds, he lays out a case for why he thinks it was actually, again, a genetically engineered construct, not, again, the result of zoonotic transfer. And so just really out of curiosity more so than anything, I watched the video, and I must say, while he didn't convince me, I think there's actually some merit to his argument. But apart from that, I think it's instructive to look at how he is laying out his case for the SARS-2 coronavirus being a genetically engineered construct in terms of our larger question that we're addressing today, is intelligent design science. Because the way that he lays out his argument and the way that people who argue it's a zoonotic origin lay out their argument actually is instructive, not only in terms of addressing the question, is intelligent design part of science, but it's also it's instructive in terms of how we, as apologists, can go about making a case for intelligent design from some features in biology or in biochemical systems. Now, before I lay out the arguments that have been presented for and against the genetically engineered origin of the SARS-2 coronavirus, there's a little bit of science that we need to present as by way of background. So if you will allow me, and this science has to do with the way in which the SARS-2 coronavirus gains entry into the host cell. Now, the SARS-2 coronavirus is a coronavirus, and it consists of a piece of genetic material. It's an RNA virus, and it's a positive sense RNA strand, a plus sense RNA strand, and it's a fairly large piece of genetic material that encodes upwards to about 20 different viral proteins. And it's encased in a protein capsid, and that protein capsid is surrounded by a membrane that is derived from the host cell that it infects. And then sticking out of the surface of the membrane are these spike proteins. That's the technical term, spike protein. Gotta love biochemistry. But these spike proteins decorate the entire surface and gives the virus this crown-like appearance, which is why it's called a coronavirus, because corona, it means crown. And so the spike proteins serve as the point of attachment for the viral particle and the host cell. And it turns out that this spike protein is what biochemists would call a homotrimer. And what that means, it means it's made up of 3 subunits. That's why it's called a trimer. Mer means subunit. So it's made up of 3 units, and homo means that the units are identical. So 3 identical subunits that interact to form a protein complex that then has as part of its architecture two domains called the S1 and the S2 domain. The S1 domain binds to a cell surface protein called ACE2, which is the angiotensinogen converting enzyme. This is an enzyme that is a protease that converts angiotensinogen into angiotensin, which is a protein that regulates blood pressure. And so this ACE2 protein decorates the surface of all kinds of different cell types, which is why the SARS-2 coronavirus can actually infect a number of different cells in the body, leading to a wide diversity of different types of symptoms that make this virus really rather insidious. But just because the virus binds to this ACE2 receptor doesn't mean it gains entry into the cell. This is where the S2 component of the spike protein plays a role. The S2 component is called a fusogen. It's fusogenic, meaning it causes essentially a fusion event which causes a fusion of the viral envelope with the cell membrane envelope, and as a result creates kind of an endosome, which is like a vesicle that surrounds the viral particle as it incorporates itself into the cell's interior. And this endosome then will fuse with things like the lysosome, which then lead to kind of an environment where the viral particle breaks down and then the RNA begins to kick in and co-opt the cell's machinery. But the point is that two domains of the spike protein have to work in conjunction, binding to the ACE2 receptor, and then that fusogenic protein has to trigger the incorporation or the endocytosis of the viral particle. Now, in order for that fusogenic event to take place, another protease on the surface of the host cell has to cleave the S2 region. And once it cleaves it, it unleashes the fusogenic activity. Now, it turns out that there are a number of possible proteases that could exist on the surface of the host cell that could cleave the S2 domain, triggering the fusogenic event. But it turns out that one of those proteases is a protease called furin, and in the S2-S1 juncture of the spike protein is a region called a furin cleavage site. Now, furin is a protease that will only cleave certain regions of proteins that have very precise amino acid sequences. And for the furin cleavage site, it's referred to as a polybasic site. And it consists of essentially an arginine residue followed by two amino acid residues that could be a variable amino acid composition followed by another arginine residue. So it would be in biochemical nomenclature RXXR, where R stands for arginine and X stands for, again, one of the other amino acids that are part of the canonical set of amino acids. So it's this polybasic site that's made up of 4 amino acid residues that the furin protein will recognize and will cleave, and this furin cleavage site exists at the S1-S2 junction for the coronavirus spike protein. Now, people have discovered that other viruses like Ebola, the influenza virus, other coronaviruses, HIV, all actually have furin cleavage sites. So this is fairly common. People have also discovered that if you modify the furin cleavage site so that one of the amino acids in the 2 or the 3 position is an arginine as well, you now have a highly efficient cleavage site that actually promotes the infectivity of viruses that again rely on furin cleavage to gain entry into the cell. And so what it turns out is that in the coronavirus SARS-2 spike protein, it indeed does have this RRAR structure, where R, again, the RR on each end of that 4 amino acid sequence, again, is part of the polybasic site, but it has an additional arginine in the 2 position and an alanine in the 3 position, and this makes it a highly efficient fusion site, which suggests to virologists that this may be part of the reason why this particular SARS-2 coronavirus is, again, so insidious in terms of its capacity to be highly infectious. Now, there's something else that's also interesting about this furin cleavage site, and that is the presence of a proline upstream adjacent to the very first arginine residue. Now, why this is significant is because proline can bind sugars. And in fact, the spike protein is called a glycoprotein because in addition to being made up of 3 protein subunits that are identical, it also is decorated with sugar molecules. And that's why it's called a glycoprotein. But proline serves as a binding site for sugar molecules, and some researchers think that this may actually create what's called a murine shield, a sugar shield that actually prevents antibodies from recognizing and binding to the spike protein, making it again resistant to the immune system clearing the virus from the body. So in other words, this particular furin cleavage site, it provides some explanation for why this particular virus is again potentially so deadly and so insidious. Now, researchers have also sequenced the entire genome of the SARS-2 coronavirus, and as part of that work have determined that it has a very high sequence identity with a bat coronavirus called the RATG13, the rat G13 coronavirus that's found in bats, a 97.4% sequence identity with this particular coronavirus. Where it differs is in 2 regions. One is in the spike protein, the sequences that are part of the spike protein that bind the ACE2 receptor are different in the SARS-2 coronavirus compared to the bat coronavirus rat G13. Now, in addition to that, there's also a high degree of sequence identity with the penguin coronavirus, with a coronavirus recovered from penguins, which leads some people to think that this coronavirus originates first in bats and then also spent some time in the penguin as a host before it transferred into the human population. Interestingly enough, that region of the spike protein, the S1 region that binds the ACE2 receptor, has a close identity to the SARS-2 coronavirus. And so this is why people think that, again, this virus originates through zoonotic transfer. Now, the thing that's interesting, however, is that neither the penguin coronavirus or the bat rat G13 coronavirus have furin cleavage sites. In fact, when you align the genomes of these 3 viruses, the SARS-2 coronavirus with these 2 coronaviruses from the penguin and the bat respectively, you note that that looks as if this furin cleavage site along with the proline residue have actually been inserted into the SARS-2 coronavirus. It looks as if there was some kind of insertion event. And this insertion is at the right place in the S1-S2 juncture for it to be cleaved by furin and to trigger its fusogenic properties. And again, that proline residue is giving it the just right properties that very well may shield the spike protein epitopes from antibody binding. And so, again, this is what I present to you is essentially facts. Now, there are furin cleavage sites that appear at the S1-S2 junction in other coronaviruses, but the coronavirus that has the closest sequence similarity to the SARS-2 coronavirus with a furin cleavage site is a virus that has only a 37% sequence identity. So in other words, when you're looking at the phylogenetic trees of these coronaviruses and you're looking at the different coronavirus families, you have the SARS-2 coronavirus that clusters over here with the bat and the penguin coronavirus forming part of a larger family. SARS and the MERS coronavirus appear over here as well. And then you have over here, again, coronaviruses that are in a completely distinct grouping that have furin cleavage sites. But if you mapped out the SARS-2 coronavirus with respect to its furin cleavage site, it would map over here. It would map over here. So these are all well-established facts. And now this is where, again, the two different groups construct their arguments for and against the origin of the coronavirus being either in a laboratory setting or being, again, as part of a natural reservoir that through zoonotic transfer was introduced into the human population. So let's take a look at the argument now that Dr. Chris Martenson presents. And it all centers around, again, the nature of this furin cleavage site, because he basically argues that what we're looking at here is a really odd situation where you have this cleavage site that shows up in the SARS-2 coronavirus seemingly out of nowhere. And his argument is this, that it'd be one thing if there was actually sequences in the region where it looks as if the SARS-2 coronavirus has the furin insert. It'd be one thing if there were nucleotide bases in that region of the genome that corresponded to nucleotide bases in the bat coronavirus and the penguin coronavirus that then underwent single point mutations to create this furin cleavage site. But the fact that it's an insert seems really very odd. It almost looks as if it was artificially inserted into this particular region to make this virus deliberately much more infectious and to gain much more readily entry into the host cell. And he argues that, again, if it was a series of single polynucleotide polymorphisms that created the site, that would be, again, an argument for its zoonotic origin. But the fact that it looks like an insert is highly suspicious to Chris Martenson. I'm sorry. And on top of that, he also argues that virologists have been studying this particular cleavage site since 2006 because, again, it's very important for the infectivity of the Ebola virus, influenza, and other HIV, other viruses like that. And in fact, researchers have been doing these gain of function studies using these furin cleavage sites and artificially inserting them into a whole range of different types of viruses to understand the role that furin cleavage plays, again, in the process of cell entry of the viral particle into the host cell, as well as deliberately trying to, again, increase the infectivity and the virulence of a number of viruses in a laboratory setting. And on top of that, he points out that in 2019, there was a study done by a team from Beijing, China, that did this very experiment where they intentionally genetically engineered a chicken coronavirus, inserting into it, again, this furin cleavage site into its genome. On top of that, he argues that the likelihood of an insert is a highly improbable event, that while single polynucleotide polymorphisms, these single point mutations being introduced into a viral genome is a rather commonplace occurrence. The introduction of inserts is a rather rare mutational event, and the fact that this mutational event occurred in the just right location with the just right set of sequences to make it, again, this highly infectious agent, to him seems to defy a natural process explanation. So on the basis of these 3 points, the artificial appearance of the furin cleavage site insertion, the fact that it's highly unlikely to have occurred through some kind of evolutionary process, and the fact that researchers have been doing these furin insertion types of studies suggests to him that this was actually a virus that was created in a laboratory where maybe these researchers in the Wuhan Institute were actually working with this particular bat coronavirus, or a coronavirus from penguins that, again, where they did genetic modifications to make it, again, more infectious as part of a gain of function type of research studies, and this virus somehow got out of the lab, and as a result of that, began to infect people. So that's his argument for it. And now, I must say, I don't know that I'm completely persuaded by his argument, but I do find the argument to have some kind of merit. It's not a completely unreasonable argument that, again, Chris Martenson presents. Now, what's the argument against this being a laboratory construct? Well, this is the argument that it was presented in the paper published in 2019 at the end of the year in Nature Medicine by this international team of collaborators. And the first thing that they point out is that, again, you have this high degree of genetic similarity between the bat coronavirus rat G13 and a coronavirus found in penguins, that when you look at, again, the sequence similarity and the fact that you see the primary difference between the bat coronavirus and the SARS-2 coronavirus being in the region where you have the receptor binding portion of the S1 component of the spike protein, and that you also see, again, a much more compatible receptor binding sequence in the penguin compared to the SARS-2 coronavirus, that this gives you a very nice, clean evolutionary history, evolutionary pathway. And these researchers point out that we have observed insertion events taking place, introducing nucleotide sequences into viral genomes, and we have observed furin cleavage sites being inserted into viral genomes through mutational events. So they're arguing that this is not necessarily out of the question. However, they do acknowledge that it actually requires a sequence of highly improbable events, where again you would have an insertion event, which again is improbable, where that insertion event is introducing, again, a furin cleavage site along with a proline residue right adjacent to it, that again is a highly efficient polybasic site that again gives this virus a high degree of infectivity, as well as creating this murine shield. They argue that it also would have had to happen in a virus that had an S1 region that would be amenable or very close in terms of its sequence to being able to bind to a human ACE2 receptor or another type of human receptor protein on a host cell surface, and that it also had to be in an animal that would have cells that would be compatible to some degree with human cells. And so it's a highly improbable sequence of events that need to take place for this furin cleavage site to be introduced into the SARS-2 coronavirus. So these researchers readily acknowledge that point, but they argue that this means that you would indeed have to have this mutational event taking place in a high density viral population, which of course is exactly what you have in Wuhan at these wet markets, where you would have a high density population of viral particles that could make the likelihood of this highly improbable event take place. And then finally, they point out that, look, if you were going to genetically engineer a virus that would infect humans, that you would probably use a very different type of nucleotide sequence in the S1 region of the spike protein to serve as a receptor binding, to serve the role of receptor binding. Because there have been computational studies done by people trying to figure out how to modify the S1 region of the SARS coronavirus to be, again, to serve as an ideal binding point for the ACE2 protein. And they come up upon sequence combinations that are very different than what you see in nature. So they would argue that the sequence combination that you see in nature for the SARS-2 coronavirus is not what you would expect to see based on these computational studies. And they also argue that there's other genetic features like the genetic backbone that we see in the SARS-2 coronavirus, which is very different than the genetic backbone of the coronaviruses that have been typically studied in a laboratory setting. So on the basis of this, they argue against the SARS-2 coronavirus having an origin in a laboratory. But note, again, that their argument is constructed around, again, looking at, does this coronavirus have features that reflect the work of a designer? Can natural process explanations explain the origin of these genetic features like the furin cleavage site? And again, uh, does the laboratory studies on this, are those consistent with what we see in the SARS-2 coronavirus? So in other words, each group or each perspective is arguing using the same kind of reasoning. Does the coronavirus have the appearance of design? Yes or no? One group says yes because of the nature of the furin cleavage site. The other group says no because of the sequence identity to the bat coronavirus, rat G13, and the penguin coronavirus. Can you explain the origin of that furin cleavage site through natural process mechanisms? One group says no because it looks like an insert. The other group says yes because we have seen inserts in nature, and if you have a high density population of viruses, you could get that kind of mutational change. One group says, with regard to, again, the work that's been done in the laboratory with gain of function work and work on furin inserts, one group says, look, we see this kind of work happening in the laboratory. That further supports the fact that this has a genetic origin. The other group says, yeah, we do see that kind of work in the lab, but the features that we see are inconsistent with it being generated in a laboratory. In other words, both groups are using the same kind of reasoning to construct their argument. They're using the three distinct criteria, and they are assessing each criteria differently as they're making their argument. And I must say that the argument for the virus having a zoonotic origin, to me, is compelling also. And so I'm not honestly quite sure what I think now with regard to the origin of the SARS-2 coronavirus. But to me, the larger question that I have here is not one where I'm interested in fueling any kind of controversies one way or the other. It's more the reasoning that's being used to draw conclusions about whether or not this virus was genetically engineered or whether it was originating through natural process mechanisms. And I've been spending a number of years actually looking at the question, is intelligent design science? And I'm of the opinion that it is, that intelligent design is rightfully part of the scientific construct, because there are areas like SETI, the search for extraterrestrial intelligence, where researchers claim that they can examine electromagnetic radiation and determine, based on the characteristics of that radiation, if it emanated from a natural object or from an intelligent civilization. This is an intelligent design program where design detection is, again, involves a set of scientific criteria. Or archaeology, where researchers can pick up a rock and tell, is that rock shaped by natural processes, or is it shaped through the deliberate activity of an intelligent agent, a hominin of some sort? And again, this is a scientific question where design detection is part of the scientific construct. Or when we talk about the origin of the SARS-2 coronavirus, this is a scientific question. Does it have features that reflect the work of a mind, or does it have features that reflect the outworking of natural process mechanisms? So I think it's safe to say that intelligent design is indeed science. But this controversy that we're talking about, as well as work in SETI, work in archaeology, also gives us a roadmap, a set of criteria that we can use when we now argue that biochemical systems are the work of a mind, or that biological systems are the work of a mind. And again, that criteria is threefold. Does it have the appearance of design? Yes or no? If it's yes, that means you could reasonably conclude that that feature could very well be the work of an intelligent agent. Can natural process explanations account for it? Yes or no? If the answer is no, that further supports the conclusion that it's the work of natural process, that it's the work of an intelligent agent. Criteria 3, what does it take to create that feature in a laboratory? Does it require intelligent agency, or can it be explained through the outworkings of natural process mechanisms? So in other words, you have 3 criteria that SETI researchers, archaeologists, and these virologists Dr. Chris Martenson, who's a pathologist, are using in their argument for or against design. And so the point here is that not only can you argue that intelligent design is literally and legitimately part of the construct of science, but also you can use these types of studies to identify criteria that we can use to make a case for a creator. And unwittingly, over the years, as I've written books making a case that biochemical systems must be the product of a mind, I actually unwittingly have used that criteria. In the book, The Cell's Design, I make the argument in the cell's design that biochemical systems have the appearance of design. There's an artificiality to these biochemical systems in that they have features that reflect the work of human designers. And so by analogy, we can revitalize the watchmaker argument and make a case for the creator, but we're making the argument that biochemical systems have the appearance of design. In the book Origins of Life, which I co-authored with Hugh Ross, we make the point that chemical evolutionary processes cannot account for the origin of life and hence the origin of biochemical systems. And then in creating life in the lab, we show that intelligent agency plays an indispensable role in creating protocells, in creating biochemical systems. That, in other words, we see in the laboratory what it takes to create certain features in biochemistry, in living systems that we think to be the work of a designer. And so we've satisfied in these three books, these three criteria, as we've made a case for the creator. So again, whether you think the SARS-2 coronavirus is a laboratory construct or whether you think it's the result of zoonotic transfer, I leave that up to you. I really have no dog in that particular fight. I think both arguments, both sides of the argument have strong merit in their favor. And so I don't really have a perspective one way or the other. But again, what I do think is interesting is that this debate, this controversy, actually again highlights a very important point, and that is intelligent design really is part of science. And that because that's the case, when we make a case for the creator utilizing scientific evidence from biochemistry, origin of life research, and synthetic biology, we are making a scientific case, not a theological or a philosophical case. And so therefore, if people want to dismiss the argument by saying intelligent design isn't science, it has no place at the table of science, and therefore I can dismiss your argument for a creator, that rebuttal is illegitimate. And I think we can point to the illegitimacy of that rebuttal by looking at this Nature medicine paper published in 2019 that explores the origin of the SARS-2 coronavirus. I'm going to go ahead and bring this Facebook Live session to a close. I just want to remind you again, if you're watching this either as a live stream or recording, take a moment, check in, say hi, let me know who you are, where you're watching from. Take a moment to put your thoughts, your perspective on these two questions about the SARS coronavirus's origin, as well as the nature of intelligent design in the comment section. And then last but not least, please suggest topics or questions for future broadcasts. Until next time, I'm going to leave you with this final thought. The more that we know about science, the more that we have reasons to believe. God bless you. I pray that you stay safe and stay healthy and stay in good spirits. Until next time.